Hello, good morning, good afternoon, or good evening, depending on where you're located. And welcome to today's Greenstone Biosciences webinar. We're thrilled to have you join us for what promises to be an exciting and insightful session on cutting-edge research in cardiac disease modeling and regenerative medicine. My name is Todd Herron. I'm the director of the Greenstone hiPSC Biobank, and I will be your host today on behalf of the Greenstone Biosciences team. Before we begin, just a few quick housekeeping notes. This session is being recorded, and we will share the recording of the webinar with all registered attendees after the webinar. The webinar will also be available on demand from the Greenstone Biosciences website. Please type your questions in the Q&A box at any time during the presentation. We will get to as many as we can during the discussion period at the end. For those of you who may be new to Greenstone, we are a biotechnology company in Palo Alto, California, dedicated to transforming human stem cell technology into reliable, scalable platforms for research and therapeutic discovery. Our hiPSC Biobank has over 2,000 patient cell lines, many from patients with inherited and rare diseases. These hiPSCs are ideal in vitro disease models for new approach methods to replace the use of animals where possible for biomedical research and preclinical drug development projects. Our mission is to empower scientists and innovators with tools that enable deeper insights into human biology, whether that's through disease modeling, drug screening, or cell-based therapy development. On this slide, you'll see a few highlights of our current product and service offerings, ranging from PBMC blood cell collection and reprogramming to derive new iPSC lines, to patient-derived iPSCs and organoids, enabling personalized disease modeling. We also perform custom differentiations to virtually any cell type of the human body, using protocols that are optimized for reproducibility and scalability. And finally, we also offer contract research services from assay development and toxicity screening to high throughput compounds. To learn more about Greenstone's services and products, please visit our website or reach out directly to our team after today's event. Now it's my pleasure to introduce today's featured speaker, Dr. William Pu. Dr. Pu is professor of pediatrics at Harvard Medical School and director of basic and translational cardiovascular research at Boston Children's Hospital. His laboratory is renowned for pioneering studies in cardiac development, cardiomyopathy, and regenerative approaches using human iPSC-derived cardiomyocytes. His cutting-edge work continues to advance our understanding of heart disease mechanisms and identify new pathways for potential therapies. Please join me in welcoming Dr. William Pu.
Thanks, Todd, for that introduction, and thanks, Greenstone, for organizing this event and giving me a chance to share my lab's work. Just some disclosures, some related to some gene therapies that I'll just be describing in this talk. So just to give a brief overview of the lab, we study gene regulatory mechanisms in heart and vascular system development. We also study the developmental biology of heart and vessel development. And we also study disease mechanisms using both iPS cell and mouse models. And from this confluence of areas, we try to develop new disease therapeutics. So in this talk, I'll kind of cover several topics that touch on each of these areas, and a general outline shown here. So I'll start with some work we've done on the scalable and reproducible production of iPSCs. iPSC-derived cardiomyocytes. So, to study cardiac physiology and cardiac disease using iPS cells, it's really fundamental to produce cardiomyocytes. Doing that in a scalable and reproducible way is really fundamental to what we're doing. So although this part of the talk will be somewhat technical, I think it really sets the foundation for future work. And this work was all done by a postdoc named Max, who has just started his independent lab in Vancouver. So I think this paper by Sean Palasek really set the stage for high efficiency differentiation of iPS cells to cardiomyocytes using Wnt modulation. And many groups have subsequently modified this idea, including this study by Joe Wu's group. However, those of us who use these 2D differentiation protocols recognize several important challenges, including they yield relatively low number of cells. There's quite a bit of batch-to-batch variation, and the effect of cryopreservation and recovery is unclear. In fact, Joe Wu's group had reported that cryopreservation of these cells would damage some of their physiological properties. I was inspired by this work by Robert Zweigart, in which he described the three-dimensional culture of differentiation of cardiomyocytes, which improved the scalability of the process. But this and other work in this area left some other important questions, such as what are the functional properties of these suspension-cultured cardiomyocytes, and how do they compare to those that we were more used to producing in 2 dimensions? I think it's an important question about not just measuring troponin-positive cells, which has been the outcome measure for most of these studies, but the functional properties of the cells produced by each differentiation protocol often is not quantified. And if you want to use the cells in functional studies, that's pivotal to understand their functional properties. We also hope that this suspension culture would improve reproducibility and avoid well-to-well variation. And we wanted to understand the effect of cryopreservation on cardiomyocyte functional properties, since that's really fundamental to producing an efficient workflow. So we modified other groups' suspension cultured protocols using a bioreactor. And we were able to develop this differentiation protocol, which also uses Wnt modulation. And at the end of the protocol, we would freeze the cells using this controlled rate freezer. So we call these bioreactor-derived cells BCMs. And we would compare them to the traditional 2D monolayer, which was done in parallel, and we call those MCMs for monolayer cardiomyocytes. So, of course, we were able to get quite high yields from this method of differentiation. So we would get 1 to 200 million myocytes per 100 mil culture. The yield of these cells of the process was quite high, and the purity of the cells was also high. So we could get over 90% troponin-positive cardiomyocytes quite consistently. We did single-cell sequencing of the cells to characterize the types of cardiomyocytes they were getting. And basically, we found that about 90% of the cells were cardiomyocytes, and most of these had a ventricular-like transcriptome, with the remaining cells having a mixture of other types, such as atrial cardiomyocytes. And this seemed, in our hands, to be somewhat better than what we did with the monolayer cardiomyocytes. We looked at the morphology of the bioreactor-derived cells, and interestingly, they spontaneously have a rod-shaped morphology, whereas monolayer cells tend to be more circular. When we plate these cells on extracellular matrix rectangles to enhance their production of their organization of sarcomeres, we could see that the bioreactor-derived cells were able to assemble better looking sarcomeres than the monolayer-derived cells, and they were better able to fill these ECM rectangles. So that's shown here. They had a higher coverage of the rectangles, and the sarcomere organization of the bioreactor-derived cells was higher than the monolayer-derived cells. This plating these cells on these extracellular matrix rectangles is stressful, such that the monolayer-derived cells actually developed quite a DNA damage response in just from the plating, which was considerably lessened in the bioreactor-derived cells. We were very interested in the functional properties of the bioreactor-derived cells. So we used optical methods to measure both the cytosolic calcium transient and the action potential. So the calcium transient measurements are shown here, and what you can see is that this is comparing cryopreserved cells to fresh cells. And in red are the monolayer cells, and black are the bioreactor-derived cells. And it's clear that the cryopreservation, for reasons that we don't fully understand, actually improved the functional properties of the cardiomyocytes in that they increase the upstroke velocity of the calcium transient, as well as the calcium transient duration and amplitude. Also, the bioreactor-derived cells tended to have more mature calcium transients than the monolayer-derived cells. This was also true when we looked at the action potential by recording the membrane voltage. So again, the bioreactor-derived cells had a higher upstroke velocity and a longer action potential duration. And for reasons we don't understand, the cryopreservation actually seemed to accentuate the benefit of the bioreactor-derived cells. We also looked at force production in these cells using the traditional engineered heart tissue construct. So in this construct, you have posts that are flexible, and between them you have a muscle bundle. So that's shown here. The muscle bundle contracts, and because you know the stiffness of the post, you can calculate the force that the muscle bundles are producing. So we could see that the BCMs could be paced at a higher rate more reliably than the monolayer cells. And furthermore, they produced quite a bit more force than the monolayer-derived cells. We were interested in being able to scale the production of these cardiomyocytes. Unfortunately, the bioreactor, one of its limitations is that the volumes are fixed by the bioreactor container. So in order to do scalable production, we switched to a different type of system known as a spinner flask. And that's shown here. And probably the bigger benefit of the spinner flask is quite affordable. So that this spinner flask system costs about $10,000, something that every lab could buy, whereas a bioreactor is about $150,000 to $200,000, putting it out of the price range of most labs. So we were very interested in, in democratizing this process also by using the spinner flask system. We also quantified the yield from the spinner flasks, so we call these SCMs, and we found that it's generally the same as the bioreactor, and then you can change the volume in the flask so that you can culture several hundred milliliters at once, and you can see the yield does not decrease as you increase the volume. In fact, it seemed to improve the yield per mil as you increase the volume. So to summarize this part of the talk, we showed that we generated a protocol for the economical and scalable production of iPS-derived cardiomyocytes. These cardiomyocytes have more mature and more functional properties than the monolayer cells, and they're retained after cryorecovery. And the ability to produce large batches really improves the workflow and improves the reproducibility of studies using these cells. So in work that hasn't been published, Max also looked at an interesting variable, which is the speed at which the rotor turns in the spinner flasks. So the studies I just showed you used a rotor speed of 60. And I'm not sure why he studied a rotor speed of 45, but interestingly, when you do slow the rotor down, you get a quite different differentiation outcome. So using the same chemical differentiation protocol, at a rotor speed of 60, you get 90% iPS-derived cardiomyocytes. But at a rotor speed of 45, you can see you get these very interesting morphology of the suspended culture cells. And at the end, you actually get a lower percentage of cardiomyocytes, which is actually a desirable outcome if you're trying to create a cardiac organoid. So we named this configuration of the organoid at day 6 the dumbbell, and then you could see that later it makes this end of the dumbbell fuses to the main body, so we call that the fused sphere. And then at day 30 you get a sphere, but this is now a multi-lineage sphere. So this is what these look like in culture. And one thing we're often asked is how reproducible is the morphology of these organoids? And what we're showing here is a low magnification view showing that most of the organoids have the same shape, have this dumbbell shape. So here's a closer view, and here the green fluorescence is TNNI1 GFP. So marking the muscle component of the organoid. And you can see the muscle cells are concentrated on the big end of the dumbbell, and the small end is non-muscle. That's also shown here by recording calcium transients. The muscle part is having calcium transients, whereas the non-muscle part doesn't. So those of you familiar with heart development know that most of the non-myocytes in the heart come from a structure outside of the heart known as the proepicardium. So during normal heart development, these proepicardial cells will delaminate and then form an epithelial sheet that covers the heart that's known as the epicardium. And these epicardial cells undergo an EMT to make non-myocytes, such as fibroblasts or smooth muscle cells, within the heart wall. So our hypothesis was that this non-myocyte compartment was like a proepicardium, and that it was producing non-myocytes for the cardiac organoid. So we looked at some classic epicardial markers such as WT1 or cytokeratin, and we found that this non-myocyte compartment was positive for these markers. You can take these organoids out of the suspension culture system and put them in 96 well dishes, and they will spontaneously adhere to the dish through the non-myocyte end. And when you do that, you get an outgrowth of the non-myocyte compartment, which forms an epithelial sheet. So epithelial because it's expressing cytokeratin, and it has the epicardial marker WT1. So this is further evidence that this non-myocyte compartment is like a proepicardium. So if we look later in development, you can see that this, we'll call it a proepicardial compartment, will touch the muscle compartment, and then they fuse. And after fusion, you get this mesenchymal compartment around the organoid. And then within the organoid, you can see that there are also some non-muscle cells. And if you look later in development, you could actually see that some of you get growth of vascular cords, which we never saw in the structures that were cultured at the higher rotation speed. And in heart development, epicardial cells support the development of the coronary vessels. And so we think that these non-myocyte cells derived from the epicardial compartment are supporting the growth of these vascular cords. So we hope to use this model to study congenital heart disease. So GATA4 is a gene that's important for the formation of the heart. And in GATA4 knockout iPS cells form highly abnormal organoids, which are primarily deficient in the muscle compartment. So that if we look at a late stage of organoid development, you can see that there's a lack of muscle cells within the organoid. So to summarize this part of the talk, just by changing the physical conditions of the suspension culture and not adjusting the chemical differentiation cues, we're able to create a multi-lineage organoid that we think models formation of the epicardium and the non-muscle cells in the heart. We think compared to other cardiac organoid systems, this system forms spontaneously, and in one culture you'll get like several tens of thousands of organoids, quite different from the organoids that are produced in 96-well dish format. And we think that these will be useful to model heart development and microenvironmental niches within the heart wall. I'm going to switch gears now and talk about how we used iPS cells to model, to study the genetics of congenital heart disease. And this work was led by Feng Xiao, who's a senior postdoc in my lab, by the way, actively on the job market. And he collaborated closely with Xiaoran Zhang and Sarah Morton. Sarah was working with the Seidmans at the time, and she's now an independent investigator here at Boston Children's. So a fundamental question in congenital heart disease research is what's the cause of congenital heart disease? Through the efforts of the Pediatric Cardiovascular Genomics Consortium, the PCGC, over the past decade, we have come to understand that the genetic causes of about 50% of CHD. The flip side of that is that we don't know the cause of about 50% of CHD. And we hypothesize that a significant contributor to this unknown CHD is non-coding variants. So the best prior work that supported this hypothesis also came from the PCGC. And in this study, they sequenced the genomes of 750 CHD trios. They found about 70 de novo variants in each individual, which in total led to over 50,000 non-coding variants. So the challenge here is to identify the small subset of those variants that may contribute to congenital heart disease. So to address this challenge, in this manuscript, they used a statistical approach in which computational methods were used to predict putative heart enhancers, and then they calculated the burden of non-coding variants within putative heart enhancers compared to controls, in CHD patients compared to controls, and found that there was a statistically increased frequency of non-coding variants within these putative CHD gene regulatory regions. A limitation of this study was that they were only able to test 31 candidates and found that 5 of these 31 affected potential cardiac enhancer activity. So we started from that point, and we used the same data set of 750 whole genome sequence trios, which led to about 57,000 non-coding, de novo non-coding variants. And we prioritized about 6,600 of these non-coding variants based on their location near congenital heart disease genes, within putative heart enhancers, or predicted to have an effect on heart gene expression by this computational algorithm. So then we have 6,600 non-coding variants. So that's there for each variant, there's a wild type counterpart. And out of this data set, we wanted to identify the subset that affect cardiac enhancer activity, or in other words, are functional non-coding DNVs. So the traditional method to do this is to use a simple reporter assay in which you drive a reporter gene using the enhancer, putative enhancer. And this is usually done one enhancer at a time. So this is the gold standard assay is the transient transgenic assay. In cell culture, we would do a luciferase assay in like a 96-well dish format. To increase the throughput of this type of assay, people have switched to what's known as the massively parallel reporter assay, or MPRA. In this assay, the enhancer is linked to a reporter that has a sequence barcode, so that you can count the frequency of the barcode in RNA compared to in the DNA. And since each barcode is paired one-to-one to an enhancer, the ratio of the barcode in the RNA to the DNA will tell you the activity of the associated enhancer. So just to show a proof of concept experiment, we manually cloned 24 regions that were putative heart enhancers. And we transduced iPS-derived cardiomyocytes and measured the activity of these enhancers using a traditional GFP reporter versus the MPRA activity. And you could see that, that generally the MPRA activity correlates quite well with the traditional reporter assay. So then we scaled this up to the 6,600 CHD non-coding variants. We synthesized alt-ref pairs for these variants, and we put this into a lentiviral reporter library and use this to transduce iPS-derived cardiomyocytes. So this graph summarizes the results. So we identified 403 variants that significantly affected enhancer activity. So the blue variants are the ones that increased enhancer activity, whereas the red ones are the variants that decreased enhancer activity. And it was somewhat of a surprise to me that over half of the variants increased enhancer activity. The preconceived notion I had was that variants that would cause disease would predominantly decrease enhancer activity. You could see here, like if we look at the variants that decrease enhancer activity, in the ref allele, they have enhancer activity, and then you put the variant in, and that turns these regions into inactive enhancers. On the other hand, for the set that increased enhancer activity, you start with a region that did not have enhancer activity in the wild type, and you add the variant, and that creates an enhancer that from a sequence that was previously not an enhancer. So, of course, the gold standard test is not the MPRA assay. That just tells us the activity of a sequence isolated from the genomic context. We want to know what's the effect of the variant within its native genomic context. So to do that, Feng undertook a quite labor-intensive experiment in which he knocked in variants into iPS cells to create clonal lines, and then differentiated these variant mutant or wild-type cardiomyocytes, and then measured the expression of neighboring genes or the whole transcriptome using single nucleus RNA sequencing. So there were 10 candidate variants that Feng tested, as well as 5 negative control variants. And out of these 10 candidate variants, there were 4 that significantly affected the expression of a nearby CHD gene based on qPCR. So here I'll just show you an example. This is BCOR. Is the neighboring gene, which is an epigenetic repressor that's known to cause congenital heart disease. And the variant disrupts a SMAD motif and reduces the expression of BCOR. Here's myocardium, a well-known CHD gene. And variant here disrupts a TED motif and generates a new TBX motif. And as a result, the expression of myocardium goes down. Just for the sake of time, I won't go through these other 2 examples. But we're interested not only on the effect on the neighboring genes, but on the transcriptome of the entire transcriptome of the cardiomyocyte. So to address that question, Feng took these different, the 4 hits in the screen. So these are knock-in lines and a wild-type line, differentiated them into cardiomyocytes, and then did single nucleus RNA sequencing. So if you put all the cardiomyocytes together, you can cluster them like this. The wild-type cluster cells tend to cluster over here on the left of this U map. And the BCOR cells tend to cluster on the right, showing that they clearly have a distinct transcriptome from the wild type. The same is true for the myocardium cells, which tend to cluster towards the bottom right, also distinct from the wild type. And we can look at that statistically and show that each of these 4 variants significantly affected the distribution of the cell state, the iPS cell cardiomyocyte cell states. So to summarize this part of the talk, we showed that non-coding variants likely contribute to the pathogenesis of CHD. About 50% of these variants increased enhancer activity, which has some implications for how you design algorithms to predict impactful DNVs. We showed that 4 non-coding variants affect neighboring CHD gene expression and the overall iPS cell cardiomyocyte transcriptome. Now, in doing this study, we ran into some obvious bottlenecks, which really point for areas of future work. One big bottleneck is how do you definitively study a variant and show that it has an effect within its native context and do that in high throughput. So the bottlenecks to doing it are 1, creating the clonal iPS cell lines. And then a second and probably equal bottleneck is differentiating all these lines into cardiomyocytes and then assessing the effect. Because if you consider that for each variant, you probably want to make 3 lines, and then for each line, you probably want to differentiate them 3 times. That's like 10 times more work than just, you know, when you start the number of variants that you want to study. So we're still working on addressing this first problem, but we do have a solution for the second problem that I want to tell you about. So what Feng did is he took some iPS cells And he introduced a barcode into each of the iPS cells so that they're genetically encoded. Then he took this pool of barcoded iPS cells and knocked different variants into them so that you would get individual clonal lines that have a variant and have a genetic barcode. So then if we pool these iPS cells and then we can differentiate them. And in the final pool, you can tell which cell has which variant based on their genetic barcode. So then you can make your UMAP plot and deconvolute the identity of each cell based on its barcode. So we barcoded the cells by knocking an N12 a random 12 base sequence into the 3' UTR of GAPDH. Within the peak, that's sequencing peak of the standard 10x 3' reaction. And this barcode can be read in the large majority of cells. So as a proof of concept, Feng did this study in which he has GATA4 knockout cells that we know affect differentiation, GATA4 heterozygous cells, and then an unknown a CHD gene that was implicated by whole exome sequencing, but without functional data, WAC. So we pooled these 16 lines together and then differentiated them as a pool. So here's the cardiomyocyte clusters. And if we look at the 10 wild type lines that were pooled together, they all have a very consistent distribution of cardiomyocyte cell states, showing that this is a very reproducible system in which 10 different wild type lines have the same distribution of cell states. Then we look at our mutants. So the GATA4 knockout has a clearly different distribution of cell states than the wild type. And you can see the heterozygote is somewhat in between the knockout and the control. And our unknown WAC also significantly affected the distribution of iPS cell states. And that's shown here. So next we turned our attention to applying this system to variants of unknown significance. So here we started with the PCGC whole exome sequencing data, and we worked with Sarah Morton again, and she helped us identify 25 missense variants of unknown significance in CHD patients. Then Feng used AlphaFold to predict the pathogenic variants. So we had 8 likely pathogenic variants. And one of these, which we're calling B26, was previously shown by Vidhu Garg's group to be pathogenic both in iPS culture and in mice. So we then use these variants and analyze them with this multiplexed approach. So in this experiment, Feng differentiated 50 cell lines in one pool, and these cell lines included RBFOX2 knockouts, RBFOX2 hets, and then the 10 different RBFOX2 variants of unknown significance. So here's a short summary of the results. So we could group, so if we look at the knockout, RBFOX2 knockout, it's clearly different than the wild type. And if we look at our RBFOX2 variants, we could group them into the ones that look just like the knockout, so that those are in red here. So V26, again, is the one that Vidhu Garg had previously shown is pathogenic. So we're showing that it's much like the knockout. And then 2 additional VUSs, V6 and V2, are also like knockout. Then we have a bunch that over here in black that are like the wild type. So these variants had minimal effect. And then we had this interesting variant V10 that was not like knockout and not like wild type. So this is a neomorphic allele. So this shows that this system can also classify alleles based on their effect. And at the level of, we did RNA sequencing analysis of these variants, and we can show that they cluster based on their transcriptomes, again, into the ones that are like wild type, the ones that are like knockout, and a neomorphic set. And here we also have some heterozygous variants, and these also cluster separately. So to summarize, we think that this barcoded multiplexed analysis is highly efficient and reproducible. I think it gives you, it reduces the well-to-well variation, it improves throughput, and I think it likely enhances sensitivity because of the within well comparison. We concluded that the WAC mutation is likely impacting cardiac differentiation, supporting this as a CHD gene. And we're able to classify RBFOX2 variants into benign, loss of function, and neomorphic based on their impact on iPS cell cardiomyocyte differentiation. Okay, in the last few minutes, I just want to turn to a third topic, which is the use of iPS based models as well as mouse models for therapeutic development. So we have this basic model in which we like to start with our patients and either use their cells or their mutations that we edit into a wild type cell to create an iPS disease model. Then we combine that with bioengineering to create a tissue level model of the disease and understand the disease mechanism. And then from the mechanism, we hope to develop a therapy that we could ultimately translate back to patients. So in this, I may not have time to talk about all three of these examples, but I'll cover hopefully 3 examples in which we use this workflow to develop candidate therapies for these diseases. So I'll start with Barth syndrome. This is a disease of, it's an X-linked disease caused by a mutation in a gene called tafazin, which alters a mitochondrial lipid known as cardiolipin. And this causes both cardiomyopathy and a skeletal myopathy, as well as the third triad, which is neutropenia. So we, working with Megan McCain, who was a postdoc with Kit Parker's group, we created an engineered heart tissue model of this disease. So here we plated iPS-derived cardiomyocytes on thin plastic sheets, which are coated with extracellular matrix to line the cardiomyocytes up in the same direction, so that when they contract, they curve the sheet. And based on the extent of curvature, we can calculate their force production. So here's what wild type cells look like. They're moving the sheet as shown by the movement of this red tracker. And in the Barth cells, there's very little force production, and so there's little movement of the bending of the sheet. Now, if we're able to put the TAS gene back to these cells, we can restore their contractile force production. So this suggested an obvious therapy for this condition, which is TAS gene replacement, which we're pursuing using an AAV vector. And this is work that was done by Suya Wang and currently continued in my lab by Suilan Ho. So here we engineered an AAV TAS gene therapy vector. We gave it to a Barth syndrome mouse model, and we showed that it both improves neonatal survival and improves cardiac contraction. So we're currently optimizing this vector and hope to complete the final dose response studies of our clinical candidate vector in the next few months. The next disease I'd like to cover is called CPVT. So CPVT is short for catecholaminergic polymorphic ventricular tachycardia. This is an adrenergically triggered ventricular tachycardia. And this work was another close collaboration with the Parker group. So Donghui Zhang did the work in my lab, and she collaborated very closely with Sungjin Park, and who did the bioengineering. So just briefly about the physiology of this disease. So normally an action potential will open the L-type calcium channel. This lets a little bit of calcium from the outside of the cell in. This activates the ryanodine receptor, which releases a large amount of calcium from the SR into the cytoplasm, and that leads to sarcomere contraction. The calcium is then cleared from the cytoplasm, mostly back to the SR by circa, and a small amount of calcium will go back through the sodium-calcium exchanger into the extracellular space. In CPVT, the basic problem is that the ryanodine receptor, which is mutated in 60% of these patients, is leaky, so that in diastole, when this channel should be closed, some calcium can exit the SR, go into the cytoplasm, and drive the sodium calcium exchange, which is electrogenic. And that can cause these after depolarizations, which if they're large enough can cause triggered activity. So to model this disease, we created an engineered heart tissue model. So here we have these same lined up cardiomyocytes, with the difference is that these cardiomyocytes are programmed with a optogenetic sensor that lets them be depolarized by blue light, and a optogenetic reporter that lets us record their calcium. So here's what the platform looks like. And if we look at each of these muscle strips, we can look at the calcium wave propagating through the muscle strip. So using this platform, we developed what we call the exercise test in a dish. So we're able to use adrenergic stimulation and increased pacing rate to simulate increased exercise intensity. And wild-type tissue continues to propagate this calcium wave even at the highest level of simulated exercise. But if you look at the CPVT tissue, the tissue works well at the low level of simulated exercise, but with higher pacing and isoproterenol stimulation, you get this rotor formation. So the rotor is the tissue level equivalent of ventricular tachycardia. So next we ask what I think is the pivotal question in this disease is, why is it that the arrhythmia is triggered by catecholamines? So at baseline, the patients are normal, but then they get catecholamine stimulation, and then they become at risk for sudden death. And to make a long story short, we figured out that the catecholamines activate the beta receptor, which then activate this kinase, CAM kinase 2. And this kinase phosphorylates the ryanodine receptor, and this phosphorylation event was already known to be pro-arrhythmic. So this combination of the phosphorylation at this residue, 2814, and the CPVT mutation together causes an arrhythmia. So to test this hypothesis, we ablated the CAM kinase phosphorylation site by changing the serine to an alanine. So by making this single amino acid change, we're able to make the tissue invulnerable to arrhythmia. So this is a CPVT containing, this tissue contains the CPVT variant, but also cannot be phosphorylated at this residue of the ryanodine receptor. And these tissues are completely resistant to reentry. So this leads to a potential therapy if unmasking of the arrhythmia depends on CAM kinase-mediated phosphorylation. If we were to inhibit CAM kinase, then we should be able to protect the patients from the disease. So there's a peptide that is a extremely potent inhibitor of CAM kinase. And so our idea, when we treated our tissues with this peptide, we're able to either reduce or completely prevent their vulnerability to reentry. The reentry is shown here in the red. So we wanted to test this out in vivo. So this is work done by Vasilios Bezarides, who's now an independent investigator here at Boston Children's. So he used AAV to target this inhibitory peptide to cardiomyocytes. And this was important because small molecule attempts at inhibiting CAM kinase have failed because CAM kinase is essential in other tissues, particularly in the brain. So using AAV to target the CAM kinase inhibition to cardiomyocytes would, we thought, would enable us to inhibit it in the heart, but leave it untouched in other tissues. So to test this, we developed this AAV in which we use a cardiac promoter to express AIP in the heart. And here's a CPVT mouse under control treatment. We do ventricular stimulation and it develops ventricular tachycardia. Whereas if we treat with this AIP CAM kinase inhibitor, we can prevent the development of ventricular tachycardia. And this was statistically significant as shown here. Okay, so finally, I'd like to talk about one last disease, although I'm going to have to rush through this due to time limitation. So this condition is calmodulopathy. This is an ultra-rare dominant missense mutation in the gene encoding calmodulin. It was only recently described that these mutations would cause arrhythmia, and this is an ultra-rare condition. So calmodulin, of course, we all know it as a calcium binding protein. It mediates many calcium-dependent events, and mutations in calmodulin cause arrhythmias, and these arrhythmias are very difficult to manage with our current therapies. So this shows that actually calmodulin protein is encoded by 3 different genes, CALM1, CALM2, CALM3. So these 3 genes encode the identical protein. And the variants shown here are all calmodulopathy causing variants. So calmodulopathy occurs in 2 flavors. There's a long QT type of calmodulopathy caused by the red variants, and the blue variants cause a different type of calmodulopathy, which is more like CPVT. So our hypothesis, the motivation actually for us to do this whole study was that if we could take advantage of this genetic redundancy to deplete the damaged CALM gene as well as its wild type counterpart, but the other 2 CALM genes would provide the genetic redundancy to make this therapy safe. So shown schematically here, 1 mutant copy and 1 wild type copy of CALM1. If you deplete them, the mutant copy is what's causing the disease. If you designed a therapy that would get rid of both of these, the wild type and the mutant CALM1 allele, you would prevent the disease, but CALM2 and CALM3 would continue to express and prevent any problem from the depletion of CALM1. So we developed iPS cell lines that contain this known calmodulinopathy allele, F142L. Importantly, we also made this CALM1 allele, which is a knockout. And this would simulate the ideal complete depletion of CALM1 by a therapy. So the calmodulinopathy containing line expresses less RNA. And then here in blue is the knockout line. And this knockout line reduces the mRNA by half, probably through nonsense-mediated decay. But if you look at the protein level, the knockout line expresses the same amount of calmodulin protein, supporting our idea of genetic redundancy. So I'm going to have to speed it up now. If you look at the phenotype of the knock-in lines, they have a prolonged action potential duration, consistent with the long QT phenotype. And interestingly, the knockout line is just like the wild type, which makes sense because its calmodulin protein level is normal. So how do we deplete calmodulin in these cells? Our goal was to develop oligotherapy. And I was inspired by this study by Tim Yu, which showed that oligotherapy is a great platform for N-of-1 therapies for ultra-rare disease. So oligotherapies can deplete a gene through multiple different mechanisms, including inhibiting its processing and splicing. Can also inhibit its translation, or it can promote the transcript degradation. And there are many FDA-approved oligotherapies, although a current limitation is that oligotherapy for muscle is not so good because of cell uptake by muscle cells. In any case, we tested some antisense oligos and showed that they are able to selectively deplete CALM1 and not affect CALM2 or 3, and they also don't affect calmodulin protein level. And these therapies normalize the action potential duration, suggesting that they would correct the long QT syndrome in patients. To test this in a mouse, we obtained a CPVT flavor calmodulinopathy variant from Michael Rubart. And we tested an antisense oligo developed to the mouse CALM1. And just to make a long story short, we were able to show that this oligotherapy was able to completely protect these mice from arrhythmia. There was only one mouse that had a little bit of residual arrhythmia. And this was completely safe. It did not change the protein level, and it did not change heart contraction or the QT interval. So in summary, I hope that this work shows the power of iPS platforms to discover new therapies and develop them for translation. And I showed you 3 examples for Barth syndrome, CPVT, and calmodulinopathy. Happy to take any questions.
All right, wow, thank you, Bill. That was a wonderful presentation. It was multifaceted, ranging from using iPSCs as disease models to doing molecular genetics and even testing therapies that were discovered in vitro in vivo animal models. It's really fascinating. I wish we had another hour to listen, Bill. It's wonderful work. Now we'll move into the Q&A portion of the session. I also want to thank the over 300 attendees who tuned in today. And we'll get to some of the questions now. There's many questions in the Q&A. If you have more questions, please feel free to put them into the Q&A box in Zoom. One question, Bill, is, do you see binucleated myocytes in your bioreactor?
Most of the cells, I can't say we didn't see any, but the predominant population was mononuclear.
All right. Next question. What is your opinion on iPSC-derived cardiac progenitor cells and cardiomyocytes for transplantation, cell engraftment in a mouse model of myocardial infarction for cardiac regeneration studies?
I think that work is very exciting. I mean, I think Chuck Murray and Michael Laflamme have been leading the way. Laflamme have been leading the way in those studies, and they've gone way beyond mouse models and into primate models. And Zimmerman Group in Europe has actually put some of these EHT-like constructs into people.
Mm-hmm. All right, next question. Can semi-continuous or flow approaches improve the yield and throughput of the bioreactor differentiation process?
I'm not sure I understand that question. I mean, I think the suspension culture clearly has a better yield than than the two-dimensional system. I'm not sure what that meant with continuous flow.
Mm-hmm. All right. All right. Next question. Have you tried lower revolutions per minute, lower than 45?
So Max is continuing that work in his independent lab, and he has tried many different conditions. Um, I'm not sure all the different combinations, but he has actually, with the same chemical cues, we can even get other germ layers, such as brain derivatives, developing in these organoids.
Wow, it's fascinating how cells respond to their environment. Another question is, may I know what is the gender or sex of the cells used for the differentiations? If by chance it was female, the question is curious about the XC1 process, and will it reflect the imprinted maternal or paternal or follow the iPSC derived individuals?
Yeah, our workhorse cell line is WTC11, which is a male cell line. We did differentiate female cells as well in the system, and they worked the same, although we certainly don't have the same level of experience with them. I'm not sure about the XC11. I'm not sure what that refers to.
Right. All right, the next question is fascinating results with spinner flask cardiac organoids. I was curious to know what is driving the multilineage cells in cardiac organoids that was absent in 65 RPMs, if speed is the only factor that's changing.
Well, I think speed is reflected in my hypothesis is that it's the size of the original cell aggregate, which probably, because we're adding the chemical cues into the media, the amount that those cues diffuse into the cell aggregate is probably different. So I think, even though we didn't directly change the chemical cues, I think by changing the size of the cell aggregates, we are affecting that.
Right. Next question is, will the epicardium differentiation under 45 RPMs also happen in static culture?
Um, I guess I'm not clear on that question, because if it's static culture, it'd be 0 RPM, so then it would.
Yeah, I guess, is there a comparable monolayer epicardium differentiation
Oh,
protocol?
I see. Other groups have established both pure epicardial differentiation conditions as well as they call them epicardioids. So there's been some really nice work done on epicardioid organoids, which are made in like a 96 well format with much more complex changes to the chemical cues.
Okay. The next question is about cryopreservation. For cryopreservation in the induced cardiomyocytes, could you share some information for the detailed method, including freezing medium, freezing method, and recovery rate?
Um, yeah, so we like to use a controlled rate freezer. I think the same result could be achieved with typical cryo preservation boxes, but I think the consistency is different. Like there's more variability, so we like to use the controlled rate freezer because we think it minimizes that variability. In terms of all the details, I think we are preparing a protocols manuscript, but I think we'd be happy to share those with whoever is asking. We could email me and we could share that.
Okay, I think some of the details are also in your recent publication
Yeah.
with your communications. Further to that question on cryopreservation was, does the age of the myocytes determine the outcome for cryopreservation? In other words, does freezing younger cells have any impact versus waiting longer when the cells are more mature on the cryopreservation recovery?
So we did all of our studies at day 15 of differentiation. Whether so I guess I can't really answer whether like a day 100 cell would cryopreserve as well.
Mm. Okay, here's another question. There's a compliment, says, great work, Dr. Pooh. Two questions. The first one, have you looked at mitochondrial biogenesis and function between bioreactor cardiomyocytes and monolayer cardiomyocytes?
That's not something we've studied. I mean, of course, with our work on Barth syndrome, I'm very interested in the mitochondria, but we didn't compare between the different culture methods.
Okay, and then the second question from this participant is, in Barth syndrome rescue experiments, when did you deliver the AAV TAZ gene construct? Was it after differentiation or before?
Yeah, so those studies we published quite a while ago in 2014. And for those, we did not use AAV. We used modified RNA. And I think we first differentiated the cardiomyocytes, and then we added the TADs after we had the differentiated cardiomyocyte.
Mhm. And then another part of this question is, in mice, at which neonatal day was it given?
Um, so that depends a bit on the model. So in our TAS knockout model, there's quite a high level of neonatal lethality. And so to avoid that, we give the virus on the first day of life. It's almost a race between the rate that the virus can express and the processes that are leading to death. But we also have tissue-specific knockout models that don't have that neonatal lethality. And there we're able to even reverse an established cardiomyopathy. So if we give the virus at like 3 months of age, we can reverse cardiomyopathy that has already established itself.
All right, thanks. Here's another question. When phenotyping a causative genetic mutation for heart disease, what are the benefits and drawbacks of using cardiac organoids versus in vivo mouse models?
Yeah, this is a great question in our current day and age of the NAMs and the FDA and the NIH making their statements about in vivo models. I haven't gone completely over to using iPS cells. I still think there's a huge value in the animal models. But I think each model has its own strengths and weaknesses. So I don't think there's a blanket statement you could make about, you know, which one you should use to study a new disease gene.
Hmm. Okay, the next question is, did your lab use cardiomyocyte monolayer for action potential and other electrophysiology recordings, or is your lab using single cardiomyocyte recordings?
most of those recordings are done and we call them small cell islands. So it's not a confluent, dense monolayer, but small clusters of cells. For most of the recordings, we're using a somewhat high throughput device, the Vala Biosciences imaging cytometer.
All right, the next question starts with "Great talk, Bill," with an exclamation point. Is it possible that many of your candidate NCVs may be enriched for functional effects on gene expression pertinent to cardiac morphogenesis in cardiac progenitors rather than in cardiomyocytes?
Yeah, so we for those studies, our early time point was day 10 of differentiation, which already are, I guess you would call them early cardiomyocytes, but they're, not mature cardiomyocytes. I think that there's a lot of room for additional studies with those variants. For example, as the person suggested, studying them specifically in cardiac progenitors, or even in other cell types such as endocardial cells or neural crest cells. I think we haven't done those studies, but I think they need to be done.
All right. The next question says, "Thank you very much for the insightful talk. Could you please comment on how iPSC cardiomyocytes can be utilized for screening novel therapeutic targets in non-genetic heart diseases such as myocardial infarction?"
Yeah, so I think most of my work has focused on genetic disease because it's very easy to establish that you have a disease model because you can have you have your control and then you have your genetic variant, and you can compare the two and know that you have a disease model. In some type of acquired heart disease, like let's just say you're trying to model pressure overload heart disease, then you try to make your model in a dish, you'd have to have some way of being certain that you really have modeled that in the dish. And I think, you could perhaps use transcriptomic readouts as the way to know that you have some fidelity in your modeling, but I think it's much more challenging. Obviously, like that's the direction things need to go to apply this more widely to acquired heart disease. But I think how to design the experiments and get the right readouts is much more difficult.
Mm-hmm. Yeah, that would require simulating a heart attack in a dish, basically,
Right.
which is not easy to do. The next question comes from Germany, and it says, "Does barcoded hiPSC stay permanently inside the cells?"
Yeah, so our barcodes are in the genome, so they are permanently barcoded. So there's different ways you can do that. One is you can do the barcoding at the same time you're adding your variant edit. Or you could make a pool of barcoded cells, and then you edit the pool, and when you obtain your clone, you identify the barcode that clone has. By the way, Greenstone could make a product out of a pool of barcoded iPS cells.
Mm-hmm. Yeah, that's one approach that we like to take is clinical trials in a dish, putting multiple cell types together and then seeing how the genomic response is from multiple patients at the same time. Here's another question is, what are the most effective strategies for promoting the maturation of iPSC cardiomyocytes?
Well, one of the points of our work was that the 3D differentiation and suspension culture promotes maturation. Other groups have worked on other methods, such as using lactate or using a maturation media that might contain hormones or fatty acids to promote maturation. So I think all of these methods work, and probably they work better in combination.
Yep. Next question is, what specific type of ASO platform did you use? Did you use any specific delivery strategies for the ASOs?
So in cell culture, you can use a naked ASO. I mean, the standard modifications of ASO, and the cells will passively take those up. But it's more complicated in vivo, and as I mentioned, particularly in vivo delivery to muscle cells is a challenge. In mice, we use palmitoylated ASO, which is effective. It was highly effective for delivery to both heart and skeletal muscle in the mice. But what we're told by Ionis, who we worked with for that study, is that that's not a readily translatable modification. And so one of the barriers to cardiac ASO therapy is, what type of modification is necessary to get that into those cells in humans.
All right, thank you. Next question says, hello, Dr. Pooh. Have you looked into hybrids? I'm not familiar with those, from these iPSCs to address mitochondriopathies.
I guess I'm not sure what that is.
It's spelled CYBRID, hybrid. I guess the question is more about mitochondriopathies. Have you studied any mitochondrial-based diseases?
So Barth syndrome is a mitochondrial disease, but it's through a gene in the nuclear genome. We have not studied mitochondrial diseases caused by variants in the mitochondrial genome.
Okay, the next question is an engineering question. Can you control the CO2 and temperature of the spinner flask outside of a cell incubator?
No. So that's one of the major differences of the bioreactor versus the spinner flask, is that the bioreactor can minute by minute control the CO2 and the oxygen and the pH. Whereas the spinner flask is basically exposed to the environment of the incubator. So on the other hand, we have not found that made a big difference. So when we measured all these functional properties, perhaps the contractile strength of the spinner flask cardiomyocytes was not quite as high as the bioreactor cells, but it was a quite small difference. The other difference is that, and the reason that the spinner is actually better for the organoids, is that all of the probes that go into the bioreactor create turbulence, which causes cell aggregation when you slow the rotor down. And so actually the bioreactor does not work as well for organoids.
All right, here's another question about sarcomere protein expression. It asks, did you look at differences in expression level of beta myosin versus alpha myosin heavy chain in bioreactor versus monolayer cardiomyocytes?
If I recall correctly, we did measure that and I think it's in our paper. But I think all of the measurements we made were consistent with the bioreactor cells being more mature.
Right. Another question. You show expression of TNNI1 in the spheroids. Do you see TNNI3 or the cardiac troponin I indicating maturation?
So the reason we use that cell line is because you can buy it from the Allen Brain Institute. And immature cardiomyocytes do express TNNI1. So those cells do express TNNI3, although as you could see by their expression of TNNI1, they are continue to be immature.
All right, the next question is about CPVT. Could you get specific cell types in the differentiation, such as atrial myocytes or Purkinje cells, which have been shown to be a cellular basis for CPVT?
So we have not focused on looking at the effect of cell types, such as, like, it is very interesting, like how is the phenotype different in Purkinje cells or atrial cardiomyocytes? Those are all very interesting questions, but we haven't pursued those.
Right. Next question is, can the MPRA system detect late on-site cardiac phenotype by non-CNVs in development? PRA system.
I mean, the MPRA system is beautiful for measuring enhancer activity, or you can use it. It's basically a way to you could also use it to measure RNA stability. All of those things it could be used for.
Another question is about the barcoding experiments. In the differentiation of the barcoded iPSCs, do the cells influence each other?
That's a great question. So of course, in that system, if there was a cell non-autonomous effect, it would be masked by the mixture of cells. So that system is really designed to look at cell autonomous problems.
Okay. Next question is, are any of the iPSC lines having cardiac disease variants available to the community? So I will say Greenstone, we do have many genetic disease cell lines that are available to the academic community. And I don't know, Bill, if any of the cell lines that you have would be accessible to the community.
Yeah, the ones that we described are we're happy to distribute them. Particularly we've given many people the BARF genome edited cells, and we also have the CPVT cells. Oh, as well as the calmodulopathy cells.
All right. The next question is, how efficient is the technique of barcoding the iPSC lines?
I think the editing efficiency is very high. If I remember, it's about 60% of the cells will become barcoded.
Okay, another question about genetics is about the process for generating gene-specific knock-in and knockout lines. The question is, could you please share how these lines are typically engineered? Is it with CRISPR-Cas9 or
Yeah,
donor?
we're using CRISPR-Cas9, and we like to use a knock-in DOX-inducible Cas9, which in our hands makes the editing much more efficient.
Right. Another question is about organoids. They want to know if cardiac organoids can be incorporated into engineered heart tissue platforms.
Yeah, that's very interesting. It's something we're actually looking into, but don't have any definitive results on that yet.
Okay, another question is iPSCs in the spinner flask. What about shear stress levels and gradients from the top, middle, and bottom of the spinning?
Um, that's not something we've looked at. I guess my assumption was that the cells would stay in different positions within the flask, and so they would be exposed to all three positions.
Mhm. All right, another question is about cryopreservation. Want to know if you can speculate on why the cryopreservation seemingly improved the function of cardiomyocytes.
Yeah, well, it's a mystery to me as well. We did those experiments several times, and we compared, like, they were the same cells, fresh and cryopreserved, compared side by side. So yeah, I'm at a loss to understand, like, why it seemed to help their physiological function.
It could be a situation where the strong survive. Yes.
Well, we did measure we measured parameters such as plating efficiency, and if I remember right, our plating efficiency is about 50%. So there could be a survival bias.
Mhm. All right, another question is about extracellular matrix. Have you considered laminin 521 for your iPSC work and differentiation?
We did not test that. I did briefly read some literature about the lamins, and it seemed interesting, but we didn't test it.
All right, another question is, says fantastic talk. In the vascularized organoid models, was there evidence of macrophages?
Yeah, we did not specifically look for that, although we're currently doing single cell sequencing of those organoids. And so I would be able to answer that better in a few months.
Okay, so also, I just want to tell you there are 20 more questions. The questions keep coming in, but we're going to limit it to just 2 more questions just for the sake of time. So one question says, beautiful talk regarding the revolutions per minute in the spinner flask. Do you have an idea of whether the shear stress or the force created by the spinner flask is driving the differences in the differentiations?
I can't exclude that. It's certainly a possible factor, although as I mentioned earlier, I think the more likely or maybe more predominant factor is the size of the cell aggregates and the effect that has on the diffusion of the chemical Wnt modulators.
Okay, and then one last question. This one's interesting. Is it possible to barcode mitochondrial DNA rather than or in addition to the nuclear DNA?
Well, I think that mitochondrial genome editors have been created, and so I guess that would be possible, but we have not tested that.
All right. All right, so that concludes the question and answer session. Thank you, all the attendees, for your thoughtful and insightful questions, and apologies to the questions that we couldn't get to, just for the sake of time. Finally, I just want to thank you again, Dr. Pooh, and thank you again to everyone who joined us today for this excellent session. If you'd like to learn more about Greenstone Biosciences, stem cell platforms, reagents, or research partnerships, please visit greenstonebio.com or connect with us on LinkedIn. We will be sending out a link to the webinar recording and related resources shortly after the event. Finally, on behalf of the entire Greenstone Biosciences team, thank you all for attending, and we look forward to seeing you at our next webinar. And thank you again, Dr. Pooh.
Thank you.